Hello everyone, my name is Kazwe Patrick, a member of the Innovate, and today I'm going to discuss two extracellular depositions, that is cholesterol accumulation and amyloidosis. As we can see in the left picture, we can see the microscopic view of cholesterol accumulation in a tissue. Then in the right picture, we can see the macroscopic or gross appearance of amyloidosis in a kidney. Now let us understand cholesterol accumulation in tissues. You see, cholesterol plays critical roles in the body, of which we have bile production, the synthesis of hormones, synthesis of vitamin D3, and as well it forms a building block in the cell membranes. But its accumulation in tissues, which in this context we are referring to as cholesterol cleft, is disastrous and is usually seen as a sequel to hemorrhage. It occurs after hemorrhage. So what happens? When there is a hemorrhage, hemorrhage will trigger an inflammatory response. And during the inflammatory response, there will be macrophages that are brought into the area. So these macrophages, they will try to clean this area. They will clean it of the red blood cells that have been brought into the area. So as they are cleaning the red blood cells, they are phagocytosing them and they will break down them. The remnants will be cholesterol, which has been in the cell membranes of these red blood cells, and these will be deposited in this area and they will appear like needle-like, what we are calling acicular spaces that are admixed with macrophages. Of course, the macrophages, since they are the ones that are phagocytosing them. In this slide, we can really appreciate the organization and the appearance of these cholesterol clefts. They are microscopic, by the way. So they will appear like palisades. Palisades, simply I can refer to it as piles. There will be piles of these needle-like, clear, cleft-like spaces, and they will be admixed with macrophages. So such lesions, they will be common in the choroid plexus of old horses in the form of a tumor-like mass that is called the cholesterinic granuloma. So this tumor-like mass, it is non-cancerous, and when you look at the choroid plexus, it's a place that is highly supplied with the vascular cells, with the vasculature. It has a lot of capillaries, a lot of blood. So when an abnormal growth occurs there, it can easily rupture these blood capillaries and we shall have hemorrhage, and the same process will occur, bringing about these lesions. So in this slide, we are simply appreciating the location of the choroid plexus. It is found within the ventricles and its major role is to produce CSF. Let's understand amyloidosis, it being the second extracellular abnormal deposit. In this context, we shall refer to it as systemic accumulation of amyloid. But what is it really? So, amyloidosis, it is a disorder that is characterized by extracellular deposits of misfolded protein, which we call amyloid, that aggregates to form insoluble fibrils, and these fibrils will lead to tissue damage and they will later on compromise function. So this amyloid, it is anamorphous, which we can as well know as abnormal. It does not have the right form. So it's an amorphous proteinous substance that is deposited between the cells of many tissues. So in its structure, it is made up of glycoproteins and fibrils of indefinite length. And the protein component is made of the beta-pleated sheets. So these beta-pleated sheets, they are an abnormal configuration of proteins. Usually, the normal proteins should have the alpha helix. It's the configuration. But these amyloid, they have an incorrect configuration. That is the beta pleated configuration. And it is the one that is responsible for the distinctive stain with Congo red. When we stain them with Congo red, they will pick it and they will show and an observation can be seen that they have stained with Congo red. As I have mentioned, the depositions will interfere with the normal cellular functions of tissues. So here we can appreciate the pathogenesis, and then here in the left corner we can see the configurations. The beta pleated sheets will look like that, and then the alpha helixes, which are the normal configurations of protein, are like this. So the amyloids, they usually take up the other structure of the beta pleated sheets. In simple terms, the pathogenesis is that when you look at these misfolded proteins, they have a normal degradation. And normally, intracellular, it will be by the proteasomes, and then extracellular, by macrophages. But this amyloidosis, for it will arise when the normal control mechanism fails, and then what will in turn come up is the accumulation of these misfolded proteins, the amyloid, outside the cell, leading to amyloidosis. So now let us classify amyloidosis, and we shall base on the protein involved. And for us, remember, we are looking at the systemic amyloidosis. So the first division or class is the systemic primary amyloidosis. So this one will come into place when there is abnormal production of antibodies in the bone marrow by the B cells, and these antibodies, they will be containing proteins of a beta pleated sheet, which we saw that they resist being broken down. So these proteins will be called the AL amyloid, and these are usually produced by malignant neoplasms such as plasma cell myeloma. So this one will affect the bone marrows and to lead to production of this AL amyloid. And we should keep it in note that this type of amyloidosis is rare in animals. So systemic secondary amyloidosis is also referred to as reactive systemic amyloidosis, and for it occurs in the cases of prolonged chronic inflammatory or infectious diseases. In simple terms, it occurs where there is chronic tissue destruction. And what will happen in this condition? Usually in this condition, the protein that is produced is a protein. So this protein usually comes into place when there is a chronic tissue destruction or an inflammation. The liver produces this serum amyloid A protein, and this protein, when it is in excess in the body, what will happen is that it will be converted to A protein, and then this A protein will begin to be deposited in the tissues such as the kidney, the heart, the spleen, the liver, and the lymph nodes. Now, in the glomeruli, simply the depositions affect the mesangium and the basement membrane. The mesangium, they are specialized smooth muscle cells that will occur within the glomeruli, and their role, along with the basement membrane, they will control the filtration slits, the size of the filtration slits, preventing macromolecules from escaping into the glomerular filtrate. But these deposits, they will encroach on the capillary lumens, having affected the mesangium and the basement membrane, and they will allow the proteins to spill into the glomerular filtrate, leading to further disastrous effects. Then when you look at the spleen, for it is the most constant site for this type of amyloidosis, and the deposits will occur in the red pulp and the periarterial lymphoid sheets, and then the central arteries. In the birds, the hepatic amyloidosis is common, and it will occur in the space of DC. So that space of DC is that space between the hepatocytes and the sinusoids, and that space is very important because it's where exchange of materials between the sinusoid and the hepatocytes occur. So when it is affected, we shall have death in the hepatocytes because they will not be nourished. In this section, we are going to appreciate the differences between these AL and then the AA, these proteins, these amyloid proteins. So the AL, for it is a complete immunoglobulin light chain, or it can be amino-terminal fragments of the light chain, or both. But when you look at the AA, for it is a non-immunoglobulin protein, and then it is synthesized in the liver. Usually when there is no inflammation or these chronic infections, it is there but in small quantities. So these chronic infections, they trigger it to be produced in large amounts, and then those large amounts, the body has to eliminate them, and in the way of removing them, they end up being deposited in tissues. And this AA, as we saw, it is a precursor of the protein serum amyloid A protein, which is produced in the liver. And it is produced as a result of inflammatory state. So it has an increased synthesis in the inflammatory state. So let's look at organ involvement. So these primary amyloidosis and secondary, which organs are usually associated with them? So we cannot have a consistent or distinctive pattern of amyloid deposit that, yeah, when it is primary, it will only be deposited in this. No. But there are those organs whereby these type of amyloidosis are predominant. And when we look at primary amyloidosis, we can see it in most predominant in the heart, GIT, the respiratory, the peripheral nerves, the skin, and the tongue. While the secondary amyloidosis, it will be predominant in the kidney, the liver, the spleen, the lymph nodes, adrenals, and the thyroid. And since in this case for us, we are focusing more on the animal side, and we know that primary amyloidosis is rare in the animal side, we are going to see some of the general representations of this secondary amyloidosis in some of those organs in the next slide. This slide is showing us the glomerulus of the kidney, and we can see that in this first part we have the mesangial cells, those red cells. Yes, so these cells, as I explained before, they are specialized sematic cells, and they help along with the basement membrane to regulate the filtration slits. But when depositions occur, and we have lost these mesangial cells, these slits will be widened, or they will be stretched, and these macromolecules which are not supposed to be in the filtrate will end up in the filtrate. In this slide, we can appreciate how amyloidosis affects the liver. So in A, we can see that the deposition is extensive in the space of DC, causing compression and pressure atrophy of the hepatocytes. Then in B, we can simply appreciate the staining with the Congo red, and it will be showing those characteristic red appearance, and then it will show apple green birefringence in C when we use polarizing microscopy. Then lastly, we can appreciate the amyloidosis in the spleen. So in this illustration on the left, it is just showing us the depositions of these amyloidosis, where we can have SAGO where it is mild, and then LAD where it is so extensive. And in the right, we can really appreciate the histological view, where we shall have to keep into note the mottled pink staining in the splenic parenchyma. Yes, thank you very much for listening. We shall catch up in the next presentations.